A single-celled parasite lives inside roughly 1 in 3 human beings on this planet. It quietly infects birds, sheep, mice, dolphins, kangaroos. It can change how a rodent reacts to the smell of a cat, and yet there is exactly one place on Earth where it is allowed to have sex: the gut of a cat. It doesn't just prefer cats, it requires them. Today, the world's most successful manipulator. The parasite is Toxoplasma gondii, and the strange thing about it is the split in its life. In its body cells, it is one of the most generalist parasites on Earth. It will live almost anywhere with a nucleus and a warm body. But in its reproductive cells, it is one of the most specialist parasites we know. Host, generalist, specialist, sex. That contrast is the whole episode. Here is the cycle simplified. Cats shed microscopic eggs called oocysts in their feces. A rodent, or a bird, or a person eats something contaminated. The parasite invades, multiplies, and seeds the body with dormant cysts. A cat eats the rodent. The cysts wake up in the cat's gut. They mate. New oocysts. Cycle closed. 2 hosts, 1 cycle, 1 terrain, the cat, where the whole thing reproduces. So, the obvious question is, why? Why does a parasite that can colonize almost any warm body draw the line of felines for reproduction? For decades, this was a black box. We knew it was true. We did not know why. And then, around 2019, current research finally identified the molecule that explains it. The answer is not behavioral. It is not anatomical. It is biochemical. And once you see it, you cannot unsee it. Most mammals carry an enzyme in their intestine called delta-6 desaturase. Its job is simple. It takes a common dietary fatty acid called linoleic acid and converts it into other molecules the body needs. Cats, all felines, big cats included, do not make this enzyme in their gut, so linoleic acid does not get processed. It accumulates. The cat intestine becomes, chemically, a pool of linoleic acid that no other mammalian gut on Earth maintains. And that pool is the signal. When Toxoplasma senses the buildup, it switches programs from asexual division to sexual development. No linoleic acid pool, no sex. Current research showed this by giving rodents an inhibitor that mimicked the cat condition. The catty mouse experiment worked. Inside that mouse, Toxoplasma produced oocysts in a host that should never have allowed it. This is the first defined molecular mechanism for species specificity in any eukaryotic parasite's sexual cycle. But the cat-only sex cycle is only one of 3 plays. Inside the intermediate host, let's say a rodent, the parasite slows down and forms tissue cysts called bradyzoites. They sit in muscle and brain, dormant, sometimes for the life of the animal. They wait. And in many infected rodents, the parasite also appears to alter neurotransmitter profiles in the central nervous system. Current research shows that Toxoplasma cysts can produce tyrosine hydroxylase, an enzyme involved in dopamine synthesis. Infected rodents lose part of their normal aversion to cat odor. They get caught. The cat eats the rodent. The cycle closes. The parasite reaches the only terrain where it can reproduce. Now, a careful note. The rodent story is well supported. The human story is more cautious than the headlines suggest. Toxoplasma is associated in current information with subtle behavioral correlations in humans, slower reaction times, certain psychiatric correlations, but the data are mostly correlational, not causal. We need more than associations to make strong claims. The biology is real, The cat-mouse manipulation is real. The "Toxoplasma made me do it" headline is not where the evidence sits today. Look at what this parasite is actually doing, and you start to recognize the patterns. A molecular Trojan horse, Toxoplasma glides into a macrophage, an immune cell built to destroy invaders, and reprograms it into a transport vehicle. Sleeper agents, bradyzoite cysts, dormant in muscle and brain. Now, ready to reactivate the moment the host's immune system weakens. A single terrain supply line, a sexual cycle that depends on one chemical condition found in one lineage of predators. The Greeks built one wooden horse once. Cold War intelligence services rediscovered the sleeper cell doctrine in the 20th century. Generals have written about supply lines for 2,000 years. But here's the inversion. Whatever military strategy humans have developed, nature evolved and perfected it millions of years ago. So, what does it actually take to understand Toxoplasma gondii? It takes parasitology to map the life cycle, biochemistry to find the missing enzyme, cell biology to follow the parasite into the macrophage, neuroscience to read the dopamine signal in a rodent brain, evolutionary ecology to ask why a parasite would commit to a single predator lineage, immunology to see how the cysts hide, and, uncomfortable as it is to admit, military theory to recognize that nature was already doing all of this long before human armies had a vocabulary for it. A parasite is not a problem to be eliminated. It is a strategist that helped write the rules of the ecosystem, and sometimes the rules of human history. Your turn. Which of Toxoplasma's 3 plays surprises you most? The cat-only sexual cycle locked to a single missing enzyme, the lifelong sleeper cysts in muscle and brain, or the dopamine-driven loss of fear in infected rodents? Tell me which and why in the comments. Want more strategies from Nature's Armory? Follow us and visit santiagolab.org and Renaissance Hub. Remember, be curious as children, let nature surprise you, and stay Renaissance.